Last week we had an introductory discussion on a subject that we don't hear a lot about, bioethics, but nonetheless a subject that will impact more and more and more people in the future than most any other subject. Dr. West, who is a surgeon for 28 years, is going to be our main speaker today. I'm just here to facilitate, but I do want to make a couple points as a way of introducing our subject. He and I we talk often as we prepare for these things, and we liken the subject of bioethics to the beginning of the advent of the computer. In other words, if you were to live back when the computer was coming out, there was a whole frontier ahead of this culture that people had no idea existed, that literally has changed the world. And he and I discussed today, and I think maybe this is not an exaggeration. The subject of gene editing is probably likened to the beginning of the computer era, because there is a whole frontier ahead as it pertains to this subject. All the more important for us, 2 Timothy 2:15, to study. You and I are not called to be ignorant. We're called to be people who study, to show themselves approved, to not be ashamed, and to rightly divide the word of truth. So tonight is a lot about informing you on a subject, more so than it is discussing the ethical issues and implications of the subject, though we will breach those. But this is an attempt from a pastoral standpoint to have a congregation that is well informed. If you listen to these last week and tonight, you ought to be able to have conversations with people over dinner about this subject from a standpoint of informing them or conversing back and forth on the subject that will lead in many respects to discussions of spiritual nature. Okay? All right, so Dr. West, how you doing?
Doing well.
You feel
Thank
good?
You. Mm-hmm.
All right,
fine.
so we're going to talk about gene editing, and we'll use different terms. A lot of these terms today are going to be technical. We're going to start out with some definitions, and then we're going to build on that. So take some notes. You have your handout. And if someone comes in late with somebody, make sure that they get a handout. I'd appreciate that too as well. All right? So, all right, start us off, Dr. West, and let's get some terms out of the way. Let's start with the word chromosome.
A chromosome, what is that? Well, it's the inheritance unit in a cell. It makes you, you. There are 50 million trillion human cells in the body, and each of those cells has a nucleus, excluding the red blood cells, which do not. There are 23 chromosomes in each of those nuclei. Those chromosomes are made up of DNA, which is a long nucleic acid molecule, and you've heard the word double helix. That's just the form that it takes when it is formed. And it's made up of 4 nucleic acids. We don't need to really go into that, but that's important to know that they're just 4 nucleic acids. They are the letters of creation in a sense, in that they, in the sequence that they exist is important to every organism that has existence.
All right, so in addition to chromosomes and DNA, you have RNA. So explain that to us, and then we're going to have a little video for those of us who don't understand what's going on, really basically me. An animated video is going to help us understand this because it's important to know this kind of stuff. All right, go ahead.
Next slide, please. This is an instruction manual, in essence, for your body. And that manual contains a great many letters and therefore a lot of words. There are 3.2 billion letters, and the function of less than 0.2% are known. 20,000 genes in the genome. The genome is the actual name for the series of letters that exist to develop your functions and traits that you express as a human. Next slide. The RNA is a short nuclear acid. It's similar to the DNA, but it's much, much shorter. And what that essentially is again made up of 4 nucleic acids, but these are copied from your DNA into what's called RNA, and they are templates from the various areas of the DNA that have to do with various traits and functions. And they form molecular patterns for the cell to make different proteins and other substances. So that brings us to a discussion of what is the important issue tonight. You may have seen in the newspaper or heard on the radio or television something called CRISPR-Cas9. CRISPR stands for that name that you see below, and we don't really need to repeat that because you'll never remember it. But CRISPR was discovered as a natural process in cells. Scientists knew from the 1980s, that there was something happening in bacteria that gave them defenses against viruses that attacked bacteria. And it had to do with the ability of the bacteria to break up the virus into little parts and identify those as foreign and develop mechanisms to ward off the virus infection in the future. So in 2012, through a series of many experiments and so forth, 2 independent researchers discovered that this was a process that was known for all cells. And CRISPR is an RNA molecule. Remember I said it was a molecule that's formed to be a template for other proteins and other substances. It's usually about 10 to 20 letters long, and it has a very unique sequence so that it can be placed on a piece of DNA in a matched area, so it lines up specifically with the nucleotides that we have talked about before. And Cas9 is one of many intracellular enzymes made out of proteins that can cut the DNA or do something to the DNA. And if you attach that to the CRISPR molecule, you have CRISPR RNA, Cas9 rather. It will precisely cut and allows replacement or taking out a piece of DNA from the strand. It's kind of like cut, copy, and paste. Fortunately, we have that from the computer world, and it kind of gives you a visual idea of what that particular thing does. It has been developed very quickly. Since 2012, it's gone to at the rate that now, I think they said last year there were 2,000 scientific papers written on this particular subject. And the other thing that's very interesting is that it's become a commercial product. It's very easily done. And there are labs that are creating specific enzymes, specific genes for different traits, and they will ship those to you. All you have to do is pay for them. So they're off-the-shelf products that are commercially available, including kits that your grade schools or clubs can get to do experiments that have already been shown to be successful, but it'll show people how this is done. And you can think about this as like the opportunity for new employment opportunities, just like the computer made a lot of things for people to be able to do that were never jobs before. This is what's going to happen with this industry.
Okay, let me make sure I understand this, okay. So a biology teacher, not unlike Harley here, can spend $150 in his school budget for a kit. And that kit comes to the school, and in front of the biology class, he can take this CRISPR molecule and he can place it over the DNA of?
Well, of a bacteria.
Of a bacteria.
Usually it would be a bacteria or yeast cell, because these are experiments that have been done and demonstrated to be successful. The kit would include all the mechanical things to do the job, petri dishes, pipettes, and so forth. and then it would have the substances that you need to do the job. One of the things that often is done is to take a gene that comes from a jellyfish, which causes phosphorescence. And this particular gene can then be placed in the DNA of the bacteria, and it will demonstrate that it was successful, because the the bacteria will start being seen as a green phosphorescing substance or organism.
Okay, so for $150 you can get a kit that could take the gene out of a jellyfish that makes it glow, put it in a bacteria, and the bacteria will start to glow.
Exactly. You can even do it for a rabbit.
Okay, and then you could take two genes at the same time, one that makes it glow and one that changes something else, and make the assumption that the second gene changed because the first one made a bacteria glow.
Yes.
All right, and that's how commonplace this is? At this point?
At this point.
Okay. So we're not talking about a NASA lab or the CDC in Atlanta?
No, it's not heavy lab work. In fact, one person said if this had been in existence, his whole career studying one thing would have been done in a month.
Wow.
So you can see the compression that can happen with this particular process.
Okay, so there's a distinct difference between changing the makeup of a bacteria in a lab in a middle school versus getting involved with a germline or reproductive cell. So what's the difference? What's going on there?
Well, we need to right away understand that the body has two sets of cells, in essence. Germline or reproductive cells are the ones that come from ova and sperm and ultimately involve the formation of an embryo. The rest of the cells in your body are called the somatic cells, and they make up the organs and tissues. And gene editing can be done primarily in the ova and sperm and embryos. If you liken it to publishing a newspaper, if there's an error in the typeset in the newspaper, if you catch it before it goes out the door, you can correct it. But try to correct every newspaper that's been sent out there to change an and to an or. You can't go to every newspaper and correct it. So you have to go back to the formation of the individual to adequately change the germline. Next. Another thing to talk about is ex vivo and in vivo. If you remove cells from the body, you can do things with them, and you can change the genome in those cells that you've taken out of the body, particularly, for instance, with lymphocytes. You can take a lymphocyte and put a gene in there that will allow it to attack something like a leukemia cell, and then you can put a stimulator gene in there to make it eat up 100,000 cells before it dies. In other words, that's one way to treat a set of diseases of the blood formation that is now being heavily investigated. In fact, there was a trial of treating leukemia in children who had failed from all other treatments, and they were given cells that were individually made for each of them. They took their cells out of the body, then they made them to attack the leukemia cells. And they had success rates significantly high. Unfortunately, they began to have some remissions. So this is all still experimental. It's developmental is the way you would put it. Now, in vivo means that you're trying in some way to change the cells inside the body in specific organs or tissues that are diseased. And that's much more difficult. So trying to do that will take a lot of investigation and time.
So if you had a child with an advanced stage of leukemia without much hope, you could take the T cells out of their body, you could manipulate the genetics of those cells?
Yes.
you could reintroduce them to the body in such a way that those cells would be changed to attack the cancer cells that would remain.
Aggressively attack the cancer cells.
Okay. Okay, I'm with you.
Yeah. Next. Oh, here's the video.
What is gene editing, or genome editing as it is also known, and how does it work? Let's start by talking about genes. These are sections of the long DNA molecules coiled up inside each cell of every living thing, from microorganisms and insects to plants and animals, including humans. Think of DNA as the instruction manual for an organism. In people, genes can influence characteristics such as your height and the color of your eyes. DNA is inherited from our parents. We have a combination of two sets of genes, one from each parent, brought together to produce a new set of instructions and reshuffled in each generation. But sometimes these instructions carry errors, and these can create problems. Faulty genes can cause serious illnesses. Humanity has a long history of figuring out ways to modify genes. We have been crossbreeding plants to make them better to eat for thousands of years. But with gene editing, we've made a great leap forward. This increasingly accurate new technology is faster to use, relatively simple, and cheap compared with previous methods, and promises huge benefits. In a nutshell, it works by identifying, then cutting pieces of DNA. One way of doing this uses a component known as CRISPR to pinpoint the precise DNA sequence within the gene to be altered. Then an enzyme called Cas9 snips through the DNA, changing it or allowing it to be replaced by another stretch of DNA that is introduced at the same time. This can either replace a faulty gene with a healthy one or change a gene to make it behave differently. The methods act like a find and replace for the genetic instruction manual. By making these microscopic changes to DNA, gene editing has the potential to make big changes to our lives. Gene editing is already transforming genetic research. But at this early stage of such a powerful technology, it's crucial that we weigh the potential benefits against the possible negatives. First, let's look at the positives. Gene editing could make radical improvements to human health. For example, in 2015, doctors used genome editing techniques to cure a baby girl's leukemia. In the future, we may be able to fix the gene mutations that predispose some people to cancer, or enable new therapies for HIV, or edit the genes that cause hereditary diseases. In the plant world, it could make crops more nutritious, disease-resistant, and able to grow in difficult conditions. By editing animals' genes, we could help them resist diseases. Scientists are currently working to edit the genes of mosquitoes to prevent them from carrying malaria. So genome editing has enormous positive potential. But what about possible downsides? Genes could be edited in early human embryos to alter characteristics such as eye color that have no bearing on health. This raises the possibility of designer babies. And changing an embryo's DNA would not only affect the child, but their descendants too. These same techniques could also be used to create designer pets, or develop more virulent microbial diseases. So, alongside the many benefits of genome editing, there are also some ethical and societal concerns to consider. The technology is already in our hands. Now's the time to debate how we use it, regulate it, avoid negative uses, and unlock its potential to provide life-changing solutions to some of the world's biggest challenges.
Well, I hope that made it more clearer to you. I thought it was pretty basic and gave you the information you needed. So to see this gives you the idea that your imagination, or the scientist's imagination, is the source of their science. Anything they can imagine perhaps they'll try to do. And will it be valuable, or will it be whimsical, or will it be something that perhaps shouldn't be done? And those are always concerns with science and what's happening in the world. And you need to be aware of that. Next. She talked about agriculture, resistance to disease, increased yields. They've already done that in wheat, rye, soybeans, potatoes, etc. There are certain diseases that are devastating, and this is an opportunity to get rid of them. In animal husbandry, health and size are being affected. They're changing the gene of a goat, of a particular type of goat, for more meaty goat, and an increase of cashmere wool from the goat. All the cows are born with horns. The females, the horns are treated with cautery so they will not develop, which is painful for the cow. And they're developing now cows without horns by taking the gene for horn formation out of the DNA. Mosquitoes, she talked about that. The interesting thing about that is to think about the way they want to do it. They want to block male reproduction and release these mosquitoes as males who can't cause the species to recreate and eliminate the species. And when you start eliminating species, you have to think about the consequences. Most of us don't think the mosquito has any particular value, but I think the bat likes them. So, you know, you're not sure exactly what's going to happen. Human genes, this is an interesting thing. They're working hard to use the pig to develop it as a human organ source for transplantation. The pig organs are essentially like the kidney and so forth. They're about the same size as the human, and they function well. And there's always been something in the back of medical people's minds that maybe they would be a good source for grafts for humans. But the problem is that the cross-species issue raises a lot of problems with rejection. And so if they can introduce genes that will reduce that problem, someday you may be getting an organ, not you, but the person that needs a kidney transplant may get it from a pig, which would be interesting. We already have pig aortic valves and stuff in people, so it's not that bad, much of a stretch to think it's possible.
Well, wait a minute now. Okay, so you have a pig, and you introduce a human gene to the pig so that the transplanted organ from the pig will not cause as many intense immune issues when it's transplanted.
That's right. That's called xenografting, and you know, if you think about it, going across species is probably the ultimate problem for rejection, because we already have all kinds of rejection problems that you need to have matches before you do transplantation with humans, person to person, and people are always looking for the matching organ. As a matter of fact, that causes a lot of problems. Not only are we behind in the number of kidney transplants that can be made because of lack of donors, but we also are down because the matches are difficult to make. The other thing that's happening is pigs. This is an interesting one. They've omitted a growth hormone in the pig gene, and they have produced what they call micro pigs. They're about 8 or 10 pounds, apparently. They look like about the size of a cat. So, you know, these are whimsical ideas.
But this is for pets.
Yes.
Can you introduce a micro pig with a human genetic thing and get a transplant from your pet pig?
I don't think it would be big enough to do much work for you.
For a baby it would.
Well, yeah, but it'd have to grow. That's another issue. Interestingly, human diseases, to reproduce it in an animal has always been hard, particularly, and now if they know the gene that causes a disease, they can introduce it into like a laboratory mouse and pretty much have the same disease in the mouse that's in the human. And because the mouse is lifespan is less than 2 years, they can do a lot of studies very rapidly. So that's a really good thing. A lot of people have an idea of something they call de-extinction, which means they want to use an elephant to create a woolly mammoth. And they'll take the gene from the woolly mammoth that they found frozen in somewhere in Siberia or something and try to put it into the elephant embryo and develop.
So this is like Jurassic Park.
Yes, well, I can go on with things that they can do. They can make different parts grow from one place or another, but we don't need to go into that.
I don't know, sounds kind of cool.
Okay, next slide. What are some considered human uses? Well, we just talked about cancer immunotherapy, and she said there was a child that was quote-unquote cured, and that's true. It was the first child cured with cancer immunotherapy using T cells that were taken out of the body and then introduced back in for leukemia. And this is a way to reduce rejection because those cells are all taken out and they're used in the specific patient they're taken out of. But there could be a possibility in the future of using off-the-shelf umbilical cord T cells. That means that you take the cells out of the umbilical cord, and those are potential cells that can be used in any person, and they would then be able to be changed, and you wouldn't have rejection. Interesting thought, and it's very valuable use. They're using it for infection, particularly for finding new antibiotics, and for antibiotic resistant testing, and you know that's a big problem because we are seeing so much antibiotic resistance that those are just important things to be looking at. And another good use would be a prompt viral infection. The Zika virus scare and all the problems there, that was partially used in that to quickly identify the Zika virus. And it would really be helpful if you went to the doctor with a cold or something to know pretty quickly that it was a virus rather than a bacteria. That would be really helpful. Next. The really interesting idea is to use a virus that won't harm the patient, and you put a desired gene on the virus and hope that it will infect the organ that you want it to infect and deliver that gene to that organ, and thereby using it to cure certain diseases. Sickle cell anemia, malaria, HIV, muscular dystrophy. All these possibilities are considered things that need to be investigated.
Okay, before you go on, let's pause for a second here. Okay, so far what we've done is we've introduced various ways of providing medical intervention in and outside the body for existing diseases. So what's the problem? Is there a problem with that? Well, we're going to get into that, but I want you to begin to think as Dr. West goes through the next couple of sections at what point, as God says in Genesis 11, in Tower of Babel, these people are capable of anything. Is there a line that we cross in manipulating the genetic makeup of embryos? Is there an ethical line that we should be aware of? Are we thinking about these issues at the beginning of the frontier, not after the frontier is over? And how do we gauge intervention and manipulation from not only a positive standpoint, but from an ethically negative standpoint? So I want you to be thinking about that as he goes through this next section here.
Some more applications would be forensic applications. I can't outline those because I don't know what they are yet. And they've already raised the fear of bioterrorism by gene hacking, using it as a weapon, and using it for population control. So those are all possibilities to think about. When you hear something that's successful, though, be very wary of what the report is. Sometimes they'll tell you it's the next thing to cure cancer, but the experiments haven't been done in any way that shows that it's going to be either totally successful or ultimately successful. So,
But at the same time, 30 years ago, when you read some sort of thriller novel or science fiction novel or science fiction movie, these things were like pie in the sky, but they're not pie in the sky anymore.
no, not necessarily. Not everything that was envisioned has come true, but there are always things on the horizon. Remember that one of the attributes of man is that he's always looking for the next thing. And in a way you can say, well, where does that attribute come from? I mean, I don't think a cow in a field or a horse at the track or anything has that attribute, but man does. And I say that's part of that is the evidence that we are created by God who gave us that attribute. So man uses those attributes and they are to be celebrated, but they're not to be abused.
Okay.
Next slide. What about the use in the human genome, the reproductive cells? This is a ticklish subject because there are many people that suffer from horrendous problems because of hereditary disease. And elimination of that disease would be a noble, noble task and a noble thing to do. But the problem is that there are over 1,000 known disease-causing genes. And so it's myriad. It's many, many different genes. On top of that, some are from a single gene, but many are from many genes. Cancers, they predict, come from at least 300 different genes because they're multifocal. They arise in different tissues and organs, and it isn't just one disease. Diabetes mellitus, they say 44. There's a misprint, but coronary artery disease is 30. So you think about that, and how are you going to effectively change all those genes and know what the outcome's going to be?
Okay, hold that thought right there, because I want to make sure I understand this. Like, we will often say, so-and-so has the gene for some sort of cancer, right?
Well,
Is that true, or is there 300 different?
I don't know that I can explain it in this setting because I don't have the information. But what I'm saying is that there are different cancers and they have the possibility for different genes. On top of that, the environment is important too because it affects what happens with those genes. So you may have a gene that has been shown to be associated with cancer, but you may live all your life and never have cancer.
Because there's other factors.
Right.
Subsequent genetic factors.
Right. Or it may take 2 or 3 of the genes and you only have 1 of them.
Okay, so your point is if there's subsequent genetic factors that intensify the occurrence of the disease, and you only manipulate or edit one of those factors, although it might be a primary one, it doesn't necessarily mean it's going to alleviate the cancer.
And on top of that, always remember, this has to be done to the embryo.
Yeah.
It can't be done.
It has to be done before the newspaper gets printed.
Right. So next slide. That's what I'm saying. You can't take a person who's got a specific congenital disease process and by genetic manipulation make that process go away.
So this is going to end up being a prevalent genetic screening before birth.
Exactly.
That's going to be the thing, right?
Exactly. So,
Are there risks involved with that?
well, there are risks to the embryo, I think.
Well, that's what I'm saying. Every parent, I can see the day coming when the majority of parents are going to go have their embryos tested, right?
They may, but remember, every embryo is a unique person.
Yeah,
They're
this is where we're
created
starting to
by
get into
God.
the ethics of it, yeah.
So,
Absolutely.
so what are the consequences of human genome editing? Are we created beings, or are we to be considered engineered beings? And you can say this is kind of a bridge too far to think that magically you can create an entire population that has absolutely no problems because you've gene edited them. Both the expense and the time and all these things are going to be involved in this. Next slide. So we must weigh the benefits and the risk. And, it kind of comes down to this. Is it a dream that we can get all these things done, or will it be a nightmare? Remember, any change to the DNA to that embryo will be passed on for generations because it changes the inheritance through that gene line. And on top of that, because humans live a long life, if you're trying to change a gene to change the possibility of a disease coming late in life, you have to wait 60, 70 years to see if it's effective. There's no way to really do credible experimentation to see if that's possible. And really, we really don't like to experiment with humans unless we've proven that it's going to be possibly effective. There was a case in 1999 where a young man had a disease that dealt with, that had a problem with his metabolism in the liver of proteins, and he would build up ammonia in his bloodstream. And it's a disease that in its worst form, you don't live through the first year of life. But in the second form, you can live a generally normal life if you avoid the certain proteins that cause this problem. Well, he was a young man, and the University of Pennsylvania, they decided they could possibly introduce a gene to his liver that would replace the gene that was not there that would allow him to metabolize the material. And they injected him, after his permission was given, with this material that contained the gene and the Casper system. And he went into organ failure very promptly and died. So it set the whole idea of this back from 1999 till about now. So it's 18 years. It's set it all back. But believe me, right now there are people out there working day and night to try and find places, because one of the problems with his particular case was the experimenter had a financial investment with the company that was going to make this process. So he had a conflict of interest, and he was, those are things that we have to know about. The dollars always trump the data. In other words, he ignored some animal experiments that showed that there had been problems, and he just went ahead to the human.
Let me ask you a question. Let's say that a person has the genetic propensity to have Huntington's disease, and that's going to be late onset, right?
Well, most people with Huntington's disease know that they've got it if they get tested.
They know they're going to
they're going to get it in 40, 50, at age 40 or 50.
Okay, it's going to manifest at 45 years old. Okay. Do animal embryos, they don't get Huntington's disease?
We don't know. I don't know the answer.
So you wouldn't be able to speed up the...
You might be able to take that gene and put it in and see if it didn't cause in animals. Somebody might know that. I don't know. I haven't investigated. But that's someplace that you think about, because if anybody watched 60 Minutes this winter, there was a family in South America where the gene is very prevalent, and they tested all these members of the family, and they had lined up these siblings, and they all knew that they were going to get Huntington's disease. Huntington's disease is a disease of the musculoskeletal system, spasms in the muscle and deterioration and mental deterioration. They all knew they were going to get it at age 40 or 50. So, that you really feel for that family, and maybe there's a place that it could be done, but it would have to be done to the embryo. So that's the problem. The other thing about all this is, where's the social justice? Usually, the economics make it available to those people who can pay for it. But there's going to be a huge number of people that can't. And what are they going to feel about it?
Well, on the subject of insurance, it would say if this was a proven successful, statistically dependable process, that an insurance company would be in favor of it.
They probably would, because it would save them the expense of actual
future expensive treatment.
But it would be money has to be spent up front.
Right, and then if we have government health care, then we have the whole dilemma, would that be provided for?
Right, would that be passed on to everyone who's paying taxes to support a government healthcare system. The other issues are legal issues. Who's responsible if it goes wrong? And where does that responsibility lie? And, can you actually give permission as a child of somebody that's had their genes changed? Can you give permission? You can't, because you're not born yet.
Right.
So it's really sticky, really sticky.
We're trying to undo the fact that we live in a fallen world.
Yes, exactly. And if you look at the sheet on there, you'll see that part of what I tried to explain in the first session, and I have a book that I wrote 2 years ago that if you'd like a copy, you can have it. It's, I don't have a lot of them, but they're over here. They're $10. The money goes to the Smoky Mountain Pregnancy Center. It doesn't go to me. And you can take one, just leave $10 there. It explains a lot of what we've been talking about with Christian culture and controversy over these socioeconomic problems. But what I would like to say about that particular sheet was last week we talked about a Christian worldview versus a secular worldview. And if you notice, a secular worldview is usually is played out with the idea of what's best for me and also what works. And they're always looking for what works. So we can go to the next slide. Around the world, there's a varying restrictions on this particular process. You can see the different colors. There's ban, Canada particularly, and some of the European countries. There's ban with certain lines in
The red is banned.
yeah, the purple is certain lines are banned, and then they're ambiguous in Russia and South America, and then we have some restrictive rules. One of the rules is that federal grants money cannot be used for embryonic research, and that's something that keeps people from doing it. But in England, they're allowed to do embryonic research, but they're on the verge of banning this particular research.
And that becomes an ethical issue because you're testing on viable
Yes,
embryo.
yes. And those embryos in experimentation, where you're not trying to take an embryo and put it back in a woman's womb for development, they'll be discarded or killed. So
That is an ethical issue.
it's
Okay.
an ethical issue.
All
Next slide. Okay, Francis Collins is the head of NIH, and he's the one of the people that discovered the map of the human genome, as you'll recall. He has said in 2015, this is a line that should not be crossed. And he was very definite about this. Next slide. But in China, in 2015, right after he said this, a research group implanted 82 embryos with human genes and tried to grow them in petri dishes, and they lived for a period of 2 to 7 days. Interestingly enough, or I guess you could say fortunately enough, the transplants were by and large not successful.
Okay, what you're saying is they introduced a gene that wasn't there originally.
Yes,
And they lived 2 to 7 days.
Now, the embryos they used, in all fairness to them, were not viable embryos because they were embryos that had been fertilized with 2 sperms rather than just 1. Don't get me on that because I don't know how that works, but I understand that that's not possible to be a viable person because you've got 3 sets of DNA rather than just 2. So their experiment, another wake-up call, and promptly around the world, the scientists began to hold symposia and talk about it. And just this spring, the National Academy of Science and the National Academy of Medicine had a joint statement. This was in the United States. It says, the final word is, editing trials must be approved with caution. but caution does not mean they must be prohibited. So right now, they've already softened up the thought process about human genome editing. And I think it just demonstrates that there are scientists that want to go ahead with this. Next slide. Human ambition is a powerful and addictive force. And I don't need to tell you that, I'm sure. But scientists need to resist a head-in-the-sand approach. And I found that picture, and I thought that was pretty interesting because it was written by another scientist. He was telling his fellow scientists, you need to not put your head in the sand about the issues that are involved here. You need to be honest about it and think them through and be responsible in your decision making. Next slide. Now, what about designer babies? You saw that mentioned in the little video, and it's a pretty selfish motivation for gene editing, and you don't know what the designer baby's going to look like. Next slide. Who knows what you're going to end up with. Anyway, so everybody feels there should be a ban on designer babies. The ultimate outcome of that would be eugenics. In other words, it would be the ultimate way to do eugenics, change the population of a particular group of people or country.
Would you define eugenics for define that?
Okay, eugenics means that you are looking for a way to purify a race or certain people. People have been trying to do that since they discovered the ability for genetics to be used to change outcomes for different crossbreedings. And of course, it was a very bad name for Nazi Germany because they were ultimately looking for an Aryan race that was superhuman, doing away with anybody that didn't meet their criteria, so they would have eugenics, true outcome. Really, it's interesting. I was reading recently that the eugenics movement started in Europe, but it came quickly to the United States. And in the early 1900s, there was a lot of call for restricting the immigration and using eugenics to purify the race. What they meant by that was feeble-minded people with certain tendencies or diseases, color issues, even people who had syphilis they were thinking about getting rid of. In fact, they did start to do that through the legal system, and many people who we think would have known better were very interested in doing that. In 1917, a man wrote a book, Madison Grant. It was called The Passing of the Great Race in 1916. That book outlined all this process and the importance of it to a civilization. Guess who read that book? Hitler. And he based all of his feelings on Mein Kampf on that book. It was written by an American. Can you believe that? That's history we don't know. We don't know that history. I don't understand that. Okay, now what should a Christian do with this technology? Ready to go there?
Me? Yeah, I think the first thing, the purpose of tonight was to inform and open up a dialogue. It was not to get up early tomorrow morning and write a letter to your congressman. We're not at that point. But we do need to be talking. I think we need to be believers who think. And I think we need to be thinkers who believe. And our credibility in our witness in culture is oftentimes predicated upon our understanding of the culture, and science is part of that. So inherent to this topic. It just seems to you look at it in one respect and you go, wow, there's some serious advances that could be made to ease the pain of people, no doubt about that. But then on another seemingly undefinable sort of level, there's something about it that just isn't right, and you can't put your finger on it. Are we playing God in some level? But the more you think about that, the more you think, well, sometimes isn't our medical intervention, our traditional medical intervention, playing God? So what is this whole subject? So I sort of wanted to open up a dialogue and then maybe bring it all together at the end. Anybody have any thoughts or questions or comments on the subject of gene